All right, so just a few updates on schedules and other things going on. Just do make sure that you read the announcements. I do know that they can be naggy at times, but I usually try to let you guys know if there's something due or if you need to print something. As far as our lab schedule, we're currently on schedule. So you'll notice that next week we have Amphioxus amistides in review, which is good because today we have embryology and development and the histology of muscle and nerve. I'll be covering the histology of muscle and nerve first. But, embryology is something that we're really not going to talk about in lecture so much, other than just a reference to, occasional deuterostome developmental characteristics. So our first practical is still on target for February 2nd. And I wanted you guys to notice that you have an assignment due on the 26th, which is next week. So you're probably wondering, okay, what's that assignment? So let's go to lab materials. And notice how I have a lab writing assignment there that just appeared. Okay, I'm going to be making this into an actual assignment link. I haven't done that yet. So an assignment link where you can download the file, and then when you're done with it, you can upload it onto Blackboard. I just kind of threw it up there so that you guys could have access to it ASAP. All right, so let me open that up. All right, so this is something that I started doing when I was at Cal Poly Pomona, just to give my lab students a little bit more of a grade buffer. Because back then, all I did is grade them on, all the instructors graded them just on the 300-point practicals, and that was it. So here at St. Francis, we have smaller class sizes, so I'm able to assault you with more material that I think is really going to make you succeed in this course. So basically, this assignment is going to allow you to basically review all of the major histological tissues that we've talked about, including today. So it's a problem-solving assignment, and basically you're going to imagine that you're walking barefoot across an old wooden floor. There's a rusty nail that has come through the floorboards, and you inadvertently step on it. Using your knowledge of histology, discuss what tissues are affected by this breach of your epithelial tissues. As your foot contacts the nail, describe in detail the reflex arc that would cause you to lift your foot, including specific cells discussed in lab. After your foot has been removed from the nail, also discuss how the cells of your immune system would handle the introduced pathogens. So remember how we talked about blood? Right, the different white blood cells? That's a hint, so you can get those involved with it too. So remember to emphasize the integration of all the tissues involved. So you'll write it in essay format, double-spaced, 12-point font. It's due at the beginning of lab next week. So it's an easy 20 points, okay? And it's a way to help you really think, okay, do I really understand what we've been talking about in lecture and lab? And there's really no minimum length. Just make sure that you're including enough detail in your answer, okay? All right, any questions about that? All right. And it says 600 to 800 words here. It varies. If you write something that's 500 words and just absolutely fantastic, that's great. This is just a suggestion. All right. So let's move on to the topic of today's lab, unless there's any questions so far. Oh, one thing I did want to bring up before I start on this is, are we interested in taking some field trips this semester to an aquarium and a zoo? Okay, would somebody be willing to head up the construction of a Google Calendar and share that with the class so that we could arrange? Has anyone ever done that? Constructed a Google Calendar, Doc? Okay, then you guys need to remind me to do it. It's been so crazy. So basically what we can do is we can put any scheduling conflicts that we have, like potential traveling with athletics and things like that. It would be a one-day trip, so it wouldn't be an overnighter. It would not be required for the course, because I do know that, maybe not all our schedules are going to jive together. But I think that it's really important that you guys get a chance to, see a lot of the organisms that we're going to talk about. Plus, I forgot. to let you know that I actually did, I have to exit preview here. Go back to Blackboard. Remember how I talked about how we had a vertebrate portfolio? Well, I actually posted that now so that you have the materials that you need. Okay, so vertebrate portfolio description. So that's going to give you how many examples of the different types of vertebrates you're going to need. It's going to be just like the biodiversity portfolio you guys did in Biology 110. Anybody in here not taken Biology 110 at some point? Okay. So basically it's just an assignment to go around and take pictures of things. And there are certain numbers that I'm going to put together a slide template. And so fish, slides 1 through 12, you must have slide 11 and 12 local species that you photographed. So that may be, maybe a few of us go out to Lake St. Francis and for educational purposes, maybe we catch a fish or two, right? Reptiles, we're going to have to wait a while to see any of these, right? This should say reptiles and amphibians, sorry about that. So I'll make some updates. Birds, mammals. So basically you're going to get 1 point for each organism that's identified correctly, and there's a specific order that will be on the PowerPoint slides, and it's going to be due April 27th. You have all semester to do this and to put it together, and you can show me how it's going. Follow the directions, easy 50 points. All right. Yes?
It says local, so like if we go on a management trip, like could we use pictures of that?
I'm going to say yes. Okay? It should be, if we're out in nature and you actually find something on your own, when you're out there, that's really the whole reason for it. Instead of taking a picture of them at a zoo, if it's something that you're around walking outside and take a picture of, I'll accept it. Right? Yeah. Yeah, I'll accept that too. These are all really good questions. What if it's an introduced species, but it's still out in nature here? Yeah, you're right. That's a really good question. And again, it's something fun to do. We're going to be out looking at things, especially when the weather gets better. And if we do organize a couple trips, like to an aquarium in the zoo, you can use those pictures too. Okay. So, yes?
And is that do just like the last lab we have?
Pretty much. Actually, I think this one is, I've put it in the lecture grades because I felt like lab, there's like a lot of stuff to do. I could, if I feel like that assignment is going to benefit people in lab better, I can always move it from one section to the other, right? I'm perfectly willing to do that. Any other questions? These are good questions. All right, so let's get going on the histology of muscle and nerve and then embryology. So very brief review of some of the things that we've talked about in lecture. So muscle, its function is for our movement, and it's contractible, so it contracts. Muscle terms that you need to know. Remember that Latin is for sarco. Sarco is for muscle. Okay, so anytime you see sarco, that relates to muscle. Remember, we talked about neuro, meaning nerves too, and we'll get to those today too. So sarcoplasm would be the cell cytoplasm in a muscle cell. Myofilaments, you have myosin that's surrounded by 6 actin molecules. So myofibrils, a group of myofilaments, and then sarcomeres. These are lengthwise groups of myofibrils, which are repeating segments. So we talked about these structures in lecture yesterday. We're going to talk about them again tomorrow as we continue our discussion. There's 3 different types: skeletal, smooth, and cardiac. You can see skeletal at the top here, cardiac, and then you have smooth muscle at the bottom. I just realized that Shay won't be able to see it unless I use the mouse. All right. So let's talk about the individual muscle types. So skeletal, these are the ones that you control voluntarily, right? These are the ones when you're running, you're telling them to run. They tire very easily. They are multinucleated. So these little purple flat things here, these are nuclei of those skeletal muscle fibers. Okay, so they're at the periphery below the sarcolemma, and you have very long cylindrical muscle cells, and they're associated with our skeleton because our skeleton provides the fulcrum for the muscles to attach to. All right, now smooth muscle is down at the bottom here, and these are non-striated. So look up here, see how there's like ribs up here in the skeletal muscle? You do not see ribs down here in the smooth muscle. So you don't have that regular arrangement of actin and myosin in myofibrils. So these are involuntary muscles. These are muscles that are contracting for an extended period of time. You don't have conscious control over them. So sometimes we get constrictions and contractions in our internal organs that can cause pain. So a couple different examples of this might be in your intestine. So your intestines are involuntarily moving your food through your system. And that's good, because if I had to remember that I was supposed to do that, I would probably forget, right? So other places, like the uterus, for example, prolonged contraction of uterine muscles can result in cramps, especially during menses, so menstruation, because they contract for a significant amount of time. Now cardiac, this is another one that I'm really glad is involuntary, because if I had to think about keeping my heart beating, I'd be dead, right? I can't remember too many things like that. So involuntary rhythmic contraction. So you have shorter cells. Okay, so these aren't as long, and they have a single nucleus per cell. And they have these really weird cell junctions, which is a specialized type of gap junction known as an intercalated disc. And an intercalated disc is a way to speed up the conduction of action potentials through those cardiac muscles and to allow them to contract as a unit, as a coordinated unit. And if you were to take enough cardiac muscle cells and put them on a petri dish and grow them for a long enough period of time, they would start contracting, which is kind of neat. So you'll be looking for these intercalated discs, and you can see how the nuclei look a lot different than the nuclei up here in our skeletal muscle. All right, just a few examples of different types of muscle. So I'm going to quiz you guys really quickly. What type of muscle is this? Smooth, right? How about this? Huh? How about this one? Smooth. And then this one? Cardiac. This one? Skeletal and cardiac. Okay, our skeletal muscle slide is taken from the tongue. And I might actually duck into human anatomy and maybe steal a couple other slides, because I was looking at them. They do not look that much like this. So, but you will see the striations. All right, the second topic we're going to discuss today are nervous tissues, and as we know, their function is conductivity. One neuron is equal to one nerve cell, and a nerve is a group of cells. All right, so if we look at just a simple neuron, this is from a slide known as the giant multipolar neuron, and we have a slide like this in lab, but I don't think it says multipolar neuron on it. So in this particular example, you can see the soma really well, so that's the main cell body, and you have dendrites coming out, and you have some glial cells that are associated here. There is a clearly defined axon coming out of this one, so you may or may not be able to actually tell which one's the axon, and that's okay, right? The axon is going to be the longer one, and we're also going to be looking at kind of a longer nerve cell, and you'll see myelin sheaths, which usually stain dark depending on the slide preparation. I believe ours are going to be pinker in color. But right here, those are the gaps called the nodes of Ranvier, where you have an action potential that is conducted at each node of Ranvier and skips to the next one. So it's saltatorial conduction, leapfrogging, and the myelin sheath is composed of Schwann cells that are wrapping around that axon, essentially. So you have the difference between the dendrites and the axons really is that dendrites are going to carry the action potential this way, and the axons are going to be carrying it away from the soma. So dendrites toward the soma, axon away from the soma. And then you have the axon terminal. We looked at axon terminals in lecture yesterday. So the space right before that synapse when we looked at the neuromuscular junction, right? So the very end. So the myelin sheath, this is an insulator, and again, nodes of Ranvier, gaps that speed up action potentials, and we call that saltatorial conduction, right? And just random fact, there used to be a punk band in the Los Angeles area called the Nodes of Ranvier, and I went to see them play. It was pretty awesome. All right, we're also going to look at a cross-section of the spinal cord, and I think the ones I got were pig cross-sections. There may be a rat cross-section too. You're going to have to look at them underneath a dissecting scope in order to see this amount of detail here. But you can also look at them up close as well. But if you do want to get a picture of them like this, there are dissecting scopes over at the right. So you have white matter and gray matter in a spinal cord. So you have a white matter on the outside and gray matter on the inside. The white matter is myelinated, so very fast action potentials, and the gray matter on the inside is unmyelinated and is a bit slower. So you're going to have to identify the dorsal horns, so these horns at the top, and the ventral horns. You'll have to look at the central canal. Not all of you will have a visible central canal, but there are some structures that you guys will need to know, and those are identified in your laboratory report. So I've done another PowerPoint lab report with all the labels of the structures that you need to know for all these slides, but this is a nice reference for you. All right, so this is where reflex arcs occur, so sensory and motor neurons are involved. So you have sensory information, and that sensory information is going to go up through this dorsal root, span the synapse, go into the ventral root, and then go out, and it's processed as motor information. Okay, so just think back to the person putting their hand over a flame. That information sensory is going to go up into the spinal cord, but get processed really quickly and go back out through the ventral root and tell you to move your hand away. All right, we're also going to look at the structure of specifically the cerebellum, but I'm going to talk a little bit about the brain in general. So the white matter in the brain is myelinated, and it's also got a fast action potential here, but it's inside, so it's flipped. Okay, the gray matter unmyelinated, it's slow, and it's outside. All right, so I'm going to just go back here. Okay, so gray matter inside here, white matter outside. Now it's switched. So white matter inside, gray matter outside. So your brain is separated into a couple of cortices, and those cortices are made of gray matter, and they're responsible for learning and reasoning. In mammals, we have large cerebral cortices, right? We have a cerebral cortex on each side. So the cerebrum, which is one of the main portions of your cortex, this is where complex responses occur. This is where our memories come from. The cerebellum, this is just primitive coordination and primitive responses like controlling your body's breathing, right? Controlling your heartbeats, controlling all the very primitive responses. And there was actually a chicken, of all things, that back in the early 1900s, somebody had chopped its head off, only they didn't quite cut it all the way off, and the cerebellum was still left intact. And it was kind of a sideshow freak. Instead of finishing the job, the farmer kept it and just basically fed it through the exposed esophagus and kept it alive for several years. It was really sad. But it was able to do very primitive things because it had its cerebellum. All right, we're going to focus a little bit closer on the gray matter. So what we're going to see is that there's a granular layer, which is the innermost portion of that gray matter, and it's granular because you can see a lot of different nuclei here. And then you have these very large neurons known as Purkinje cells. So they're very large and they mark the boundary between the granular layer and the molecular layer. So the molecular layer is to the outside. And you can see when you look at your slide, even just by picking it up, you'll be able to see a network that looks like a tree. It's branched, and people call it the arbor vitae, like basically the tree of life. And that's found in all of our brains, actually. It's kind of pretty. All right, the last topic we're going to look at today is embryology. So this is something, again, we're not really going to discuss that much in lecture, but we're going to focus on what happens in deuterostome development. Okay, so embryology, we start with a sperm and an egg that create a zygote, and then they start to divide, and then they start to become a big ball. The ball becomes hollow, and then gastrulation occurs where the ball pushes in and creates a new cavity. So that gastrulation is going to form a blastopore that's either going to become a mouth or an anus. So if it becomes a mouth, it's a protostome. So mollusks, annelids, most animals are in this category. But deuterostomes like us, we actually form our anus first and our mouth second. So this would become our anus, and we have a very similar gastrulation process, but the coelom does form a little bit differently. So you can see ectoderm, mesoderm, and endoderm characterized by blue, red, and yellow respectively. These are the germ layers that are going to become the different tissues in our body. So a few key terms for deuterostomes is that we have radial cleavage, and what that means is that the little blastomeres are on top of one another. Like I always think of it as a Rubik's Cube that hasn't been twisted. Spiral cleavage is like you twisted that Rubik's Cube. Okay? And we're going to talk about a few other things too. But deuterostomes, we do have indeterminate development at some points of our early embryological development. So we do go through periods where if you break off one blastomere, which is just one of these cells here, that can still grow into an entirely new organism. So that's where my twin sister and I come from, is from that occurrence. Okay, and we have mesoderm arising from these outpockets, which we'll be seeing, that are known as a coelomic sac. And it's a primitive gut outpocketing, because this gut space called the archenteron is going to become the innermost tissues of our body and our digestive tract. Okay, so a few general things about development strategies. So birds, they invest a lot in their eggs, and they invest a lot in their yolk because they do a lot of their development in the egg. Frogs have a short development, right, because they become tadpoles and they essentially continue to develop and metamorphose out of the egg. Mammals, we have a very small amount of yolk in our egg because the yolk isn't really needed, right? Just to get that egg down the fallopian tube enough for that sperm to fertilize it and then implant on the inside of your uterus, right? So there's a few terms I want you guys to know. So echinoderms, amphioxus, and us, we have a small amount of yolk, which is called microlecithal. The distribution is even, so isolecithal, and there's holoblastic cleavage. So holoblastic, meaning that they're essentially dividing evenly. Amphibians and fishes are mesolecithal, so they have a medium amount of yolk, and they have a teleolecithal type 1 yolk distribution, but they do also have sort of holoblastic cleavage that's even when those cells are cleaving. The sharks, birds, and reptiles are quite a bit different. They are macrolecithal, and that means they have a large amount of yolk. That's why we enjoy eating bird eggs so much, because that yolk is really tasty, right? Huge source of proteins and fats, and meant for sustaining the development of a growing organism. They have very uneven yolk. It's a teleolecithal type 2. If you think about it, crack open an egg, the yolk is clearly separated from the white. So in other types of organisms, you don't get that clear separation. So the type of cleavage, it's a little different. Meroblastic cleavage, we'll talk about that a little bit more, okay, as we're going through the class today. So why do we look at starfish development, first of all? Because it's so easy to see what those stages are, because you can see right through them. So just a really short video about sea biscuit. So sea biscuit is a type of echinoderm. So this is a female releasing her eggs. You can see her tube feet. So she basically releases them. There's the male releasing his sperm. So they have external fertilization. There are the sperm. All right, so now we have an egg. Notice how all of a sudden there's this development of a fertilization membrane, and you start to see something happening with the nucleus. All right, here you can see the cell spindle fibers, and we're going to wait for those to come together and form one nucleus, and then the genetic information divides and the cell is going to split. And this is sped up obviously a lot. All right, so from 4 to 8 celled, 16 to 32 cells, that is something known as a morula stage. But once it becomes open like this, it's a blastula, and it pushes in and becomes a gastrula. And then they are free swimming until they find a place to kind of go and be safe. All right. So I've put together just some examples of a nicely clearly stained specimen, and you are responsible for knowing these terms. So here you can see a clear nucleus, and a nucleolus is a dot in the center, and cytoplasm. Before that egg has been fertilized, it has a vitelline membrane that's stuck onto the surface of the egg. As soon as it's fertilized, that membrane starts to detach and it becomes the fertilization membrane. And part of the reason for that is that it prevents further insemination or fertilization by the other sperm. We call, once they start to divide, these different cells blastomeres. So you can see what happens through the 4-celled and 8-celled stage. So the 16-cell stage, the morula, is basically a ball of cells. It becomes a blastula as soon as it starts to get hollow in the center and have what's known as a blastocele. Here we have much more cells, so a very developed blastocele. And then the early gastrula shows the moving in, creating a blastopore, and that moving in of materials is known as invagination in embryology. And you can see in the late gastrula, you have that space which is forming the endoderm, and we call this space an archenteron. The blastopore, which is going to become the anus, because this is a deuterosome, and you may get to see some coelomic sacs coming out that will actually be forming your mesoderm tissue. So your mesoderm, this is the tissue that forms your skeleton muscle and the connective tissues, the peritoneum that hold your guts in place. Ectoderm, your skin, nervous system, and then endoderm, your gut and your lungs. All right, as far as frog development is a bit more complicated. So I've put together some scribbles for you guys because I like to simplify frog development as much as possible before we look at some more gory, detailed pictures. So What you have in frog eggs is you have a darker animal pole. So if you've ever seen the eggs of frogs, they have a darker pole, and then a lower pole that's lighter known as the vegetal pole. So not vegetable, so it's not animal and vegetable, it's animal and vegetal, all right? And each of these have different ultimate destinations. So the animal pole, this has less yolk, it divides very quickly. At the 4-cell stage, you can see some cleavage plates forming, and you'll often see something known as the gray crescent, which is the shaded area. At the 8-cell stage, you start to see that there's a difference between the animal pole and the vegetal pole when it comes to the size of the blastomeres. So the blastomeres at the top of the animal pole, they're small because they divide quickly, right? The faster they divide, the less time those individual cells have to grow. All right, so you start to get this division between micromeres, which is literally just small blastomeres, and macromeres, large blastomeres. All right, so these micromeres are small because they have a lot of yolk in them. The macromeres are large because they have less yolk to support their rapid development. Okay, so they develop slower. We still have a morula stage, and then there's a blastula. So here we have our animal pole and our vegetal pole. The blastocele should form near the animal pole. And then what's going to happen is you have invagination occurring similar to a starfish, but you still have this obvious remnant blastocele, and there's this really interesting movement of cells that now start to move down, and that's called epibole. And I'm going to show you guys a video of frog development, and you'll get to see what epibole actually is. By the time you get to a late gastrula, you'll have a yolk plug plugging up that blastopore. And on either side, you have the dorsal lip and the ventral lip of the blastopore. You can see the archenteron is getting bigger and the blastocele is getting smaller. All of this is yolk to help that frog start to develop. And then we have the clear differentiation of tissues, the endoderm, mesoderm, and ectoderm. So your frog slides will look something like this, okay? But because we're taking a section through the animal, it's going to be different depending on whose slide you look at. So you definitely want to look at a number of different slides. All right. All right, so let's look at what this really looks like. All right, so an egg. Here you see a cleavage furrow. You start to see some divisions happening. Notice how the cells on the top are now obviously smaller than the cells on the bottom. They have more energy to divide faster than the cells on the bottom. So right now there's a hollow space developing on the inside, and that's the blastocele. But it's going to start enfolding, and this is epibole right here, the movement of the cells down. You guys want to see that one more time? It's kind of amazing. Okay, wait for it. So as the material starts to go in to form that blastocele, the cells start to fold around that specimen. And here you can see the yolk plug. And then we haven't gotten to neurulation yet, but we will in a second, the development of your dorsal hollow nerve cord. So you can see how it's starting to develop a ridge here. Okay, and there's the development of that dorsal hollow nerve cord. It's going to fold itself like that. So it's pretty amazing. It's starting to look a little bit more like a vertebrate. Starting to show some gill slits. Starting to get a postanal tail. So you can actually see this happening in frog eggs. And then they become tadpoles. And hopefully you guys have had a chance to raise tadpoles and watch their legs grow. Has anyone not done that? Are you guys serious? You've never done that? Oh my goodness. All right, it's technically sort of mostly illegal to do nowadays, which is kind of a bummer. But I'm supposing if we could get a collecting permit or something for educational purposes, maybe we could do that. We'd have to run it through an IACUC protocol and all that. But seriously, one of my favorite things to do as a little kid was to take in little Hyla regilla tree frog species and then release them when they had developed. So these are some more realistic pictures of what you guys should be looking for. But again, you're going to get a slide cross-section. So this is A through F, and this is G through J. So these are going basically in order of development. All right. So they'll look more like your slides than what I've tried to draw for you. All right, now we're getting to neurulation. So neurulation was what we saw towards the end of that video, which is kind of the pushing in and then wrapping around to form that neural tube. So this is the formation of tissues or histogenesis, and the formation of organs, it's organogenesis. So your central nervous system starts from something known as a neural plate, which is just the layer of cells in which initially the notochord is embedded. Okay, so what's going to happen is that neural plate is going to elongate, and you'll see a groove developing. The notochord, you'll see, sort of migrates down a little bit. And then what happens is through pushing up, kind of like how this picture is showing right here, it seals it off. And so above your notochord now you have a neural tube, which becomes your dorsal hollow nerve cord. What's going on the inside is that you have further development of mesodermal tissues, endodermal arms, and as it starts to develop more, you have somites, intermediate mesoderm, and hypomesoderm that are going to develop into different parts of your nervous system and musculature. And then finally you have your neural tube and neural crest cells. So your neural tube is going to differentiate into your spinal cord and your brain, and your notochord is going to develop into vertebrae. So what we saw with frog development, early human embryos don't look that different. We look very much alike at early stages. So these neural crest cells, they're important because they are going to form Schwann cells, pigment cells, gill arches. It's going to give rise to our skeleton, tooth buds, and then different components of our nervous and endocrine system as well. And here's some images of what your slides might actually look like. So our slides, we have a lot of slides. Well, I guess we only have one slide of each of these stages, unfortunately, but they will often have many slices on the same slide. So do make sure to share them. One weird thing is that they're not called neural plate, neural groove, and neural tube. There's just early, middle, basically, and late. So you're going to have to search around the slides to find something that looks like this in all the cross sections that they've taken of the specimen. Okay. And let me know if you have difficulty doing that, because if we can't find a clear depiction of a specimen in any of the slides, then you're not responsible for it in your lab report, okay? All right. So that's basically it.